All right, welcome everybody to the review of the SAR, SAS, and Coordinated Assessment Data Exchange. I'm Meg Deloff. I work for PSMFC and PNAMP. I'm just gonna walk you guys through some logistics before we get started, in case you're unfamiliar with Teams. Next slide, Mari.
Sorry.
No, you're totally fine. Please turn off your camera and your microphone by clicking the icons on your team platform. If you're not speaking, obviously, Rich and everybody else who's talking, Casey and Mari, can leave their cameras on. It just kind of helps folks not be distracted, and we can mute you, but we can't unmute you. So if you folks have comments, want to ask some questions, feel free to unmute yourself, or you can also type in the chat. Some agencies don't allow chat, so if that happens, just obviously feel free to unmute yourself. A couple troubleshooting tips. Thanks, Mari. If your Teams is being finicky, just try leaving and joining again. Sometimes that helps kind of wake it up. Or use a different browser. Edge works better than Chrome or Firefox, depending on what you're using. And then desktop version versus browser, vice versa. So if your Teams is glitchy, you could try those things. Otherwise, that's all I've got. I'll try to triage if you guys have questions. So send me some chats if you get hung up on anything, and I'll see if I can help. Otherwise, I'll hand it over to Mari. I think you're doing agenda, or is it Casey?
I think it's Casey.
Okay.
Hi, everybody. Thanks for joining. I'll go ahead and introduce myself. I'm Casey Bleasner. I work for the Oregon Department of Fish and Wildlife in our East Section, Salmon and Steelhead Science, out of La Grande, Oregon. I also work with StreamNet and Coordinated Assessments, and am on the PNAMP Fish Monitoring Work Group as a core team member. And so I'm here along with Meg and Mari to kick off this SAR SAS task team. Mari, do you want to introduce yourself?
Sure. I'll see if I can match how well Casey introduced herself. I'm Mari Williams. I work for Pacific States Marine Fisheries Commission on the StreamNet and PNAMP projects. Been involved with coordinated assessments from back in the day when Rich was also with coordinated assessments. And I do lots of data coordination, and it's been a fun window into the salmon and steelhead Pacific Northwest world.
Agreed. So I'm just going to run through our agenda real quick, just so you guys all have a touch point of what we're doing and where we're headed. We're going to give a quick background on the SAR SAS task team, the purpose, goals, and the fish monitoring work group task team structure. We'll pull up a slide here in a minute and talk about who responded to our initial survey and indicated they wanted to be a team member or backup for an agency. And then we will also review the coordinated assessments data exchange standard development and revision process, so that those of you that aren't as familiar with coordinated assessments as Mari and I are kind of know how that process works for how we go about changing the data exchange standard. And then we will jump in, and Rich Carmichael and Tom Quinn will give a presentation about bringing this SAR SAS task team and the ISAB report review and recommendations. And then we'll have a quick wrap up. We'll review the goals and plan for the next meeting. All right, Mari.
So if you are here for the SAR SAS data review and coordinated assessments, you're in the right place. Otherwise, pick another webinar. We're here in response to the ISAB's report from last year, where they reviewed the SAR and SAS data available throughout the Pacific Northwest to inform salmon and steelhead recovery and production metrics. They included a fairly in-depth review of the data that's within Coordinated Assessments Data Exchange, and that is what we will be discussing today. This is not meant to enforce any data exchange standards on any of the other processes that might produce or hold SAR, SAS data. And so the task team goals are, we're going to review what is currently in coordinated assessments with some excellent guidance from Tom and Rich, and discuss how we can modify the descriptions or add other data fields in order to accurately describe whether the data in the exchange is SAR or SAS. I realize I'm using lots of acronyms, at least in the SAR, SAS, that's Smolt-to-Adult returns and Smolt-to-Adult survival. Otherwise, I will really try and not use acronym speech. And so what this group is going to do is develop a recommendation for modifications to the data exchange standard. And as part of the coordinated assessments process, that will end up with a couple of different reviews to the point where we will then update the data exchange standard. All right. Casey?
So we sent out quite a blitz of emails to everybody and included a survey asking for engagement and interest in joining the team. So far, this is the task team that responded. I'm Casey. Mari and I are the leads. Meg is joining us with logistics support. She leads a lot of these fish monitoring work group meetings and teams, and so she's there to help us out. We had a couple people, Mike Bannock, Brian Leth, Pete McHugh, and Matt Siskey step in and volunteer to be members of this team. And we had Jake Chambers, Tim Copeland, Rod Engel, and Joseph Feldhaus indicate their interest as backup representatives. I think at this time, it would be great if everybody that joined the meeting, if you could type your name and your affiliation into the chat. This will also help us keep track of attendance. And then did we want the folks that volunteered as members to come on and introduce themselves?
Yeah, I think that would be great. If folks that are going to be members of the task team and providing collaboration to develop this would like to turn on their cameras and introduce themselves.
We can start at the top of the list there. Mike Bannock, are you out there?
Yeah, there are a lot of folks, and I can't quite see who's in here or not. I see Pete. Pete, do you want to come off and
Pete?
And then Brian.
Yeah, good afternoon. Hopefully you can see me. First off, can I just offer a quick clarification? I believe there was a comment, one of the comment fields I included that we still needed to work out internally within CritFic whether we would commit at a full member level. So if I could put a little asterisk next to my name, I still need to work that out with Stuart Ellis and Bob Lassard and Tom Lors. So, but I think I don't know if I was the only one that completed the survey. We're definitely interested and want to be involved on some level, but just to clarify that. But yeah, so introduction wise, Pete McHugh, fish passage scientist here at CritFic, and yeah, happy to be here this afternoon.
Great, thank you, Pete. Brian?
Pete, to that, I was going to say to that end too, to everybody that is here, we're putting this task team list up here for that reason as well, so that you can see who stepped in from your organization and filled out the survey and said they'd be a member. And then at the end of the presentation, we'll have a timeline. And so we're hoping that after the presentation, after you kind of hear all of this, there'll be some time to indicate if you want to join as a team member or talk amongst your agency about who wants to be involved. And we'll give a timeline so you have an idea of how much time we're planning on this taking. So anyway, Brian.
Yeah, hi, my name is Brian Leth with Idaho Department of Fish and Game, and I oversee the hatchery evaluations for our Chinook and Steelhead hatchery program. So my focus is really on hatchery returns and our hatchery evaluations. I know there's a number of other folks on the phone right now from Idaho Fish and Game, and I think we might potentially be maybe having somebody also participate as a member to represent kind of more of the natural production side of that as well.
Great. Thanks for being here, Brian. Matt?
Yeah, hi, I'm Matt Siskey. I'm a hatchery monitoring and evaluation complex lead in Region 5 within DFW here in Washington. I've been tasked since starting this position to develop an SAR and SAS calculator for our region and for statewide use. And so I've started some of that work, and I'm excited to continue working with you all on this. Thank you.
Great. Thanks, Matt. I'm glad you're here. Okay. So we wanted to put forward the current data exchange standard development team membership. If I slip into acronyms, DDT is DES development team. So our current membership, which is flexible as folks come and go according to what we're working on, such as when we developed hatchery coordinated assessments and as separate from natural coordinated assessments. As some of the members here have explained, our jobs are often split, natural and hatchery. And there are a lot of different implications for each side of that. We wanted to put this up so that folks knew who was representing their agencies for coordinated assessments and possibly consider joining or participating as an observer, just getting more emails in your email box. But it is a great team to work with, and we've accomplished a whole lot over the last 15 or so years. So here is our data exchange standards development and revision protocol. You can either use that Exchange Tools DES link or the QR code that's on that, but I highlighted the section that we are going to be working through in this task team, and I know that nobody can read this. So where we're at is we're looking at a DES revision. We need to change the structure of the SAR for both natural origin fish and for hatchery origin fish in order to accommodate the fields that can provide identification of whether that data point is smolt-to-adult return or smolt-to-adult survival. So right now we are at where the DES development team has said, hey, fish monitoring work group, could we get a team together? And right now we have 33 people coming together to listen or talk about how we can better identify data within the data exchange so that it can be used in an informed manner, and we can do better science with it. So we are looking at a major revision. This would have like structural changes within the database. So we go through the process of having this ad hoc work group through the fish monitoring work group develop draft DES, and then we send that to the steering committee, after the DDT has approved, and then the StreamNet steering committee sends it back to the DES development team to either finalize and set an effective date or have modifications made as the steering committee sees fit. We have a really good working relationship with our DDT and steering committee, and so there isn't much sending it back. There are a lot of folks that look at it long before it gets to the steering committee. So I don't expect that there will be much back and forth with the steering committee because folks are showing up for this meeting already. So the effective date of a new or modified DES means that in 2 months, it will be the exchange standard that data coming into coordinated assessments and going out of coordinated assessments in the exchange needs to fit. So the coordinated assessments agreement is 2 months for folks to try and set up to be able to send data in the new format and for StreamNet staff to be able to accept data in the new format. And then we have better data faster. All right. I now have the absolute pleasure of introducing Rich Carmichael. Rich, I have known for almost my whole time working in the Columbia River Basin. He was for 36 years ODFW employee. He was the program director for the East Side Oregon Fish Research and Monitoring. And he has since retired and then come back to help guide other efforts as a consultant. And in his role here, he was working for the council on the ISAB, providing his knowledge and skills. Alongside Rich today, we have Dr. Thomas Quinn, and he is a professor of aquatic and fishery sciences at the University of Washington. He was a former ISAB chair, and I'm not sure if he was chair while they wrote this document, along with other support and help from folks. But we are absolutely honored to receive the very constructive criticism of how to make coordinated assessments better. Rich was part of the development of these original SAR, at least the natural origin SAR data exchange standards. And I'm glad that he has been able to circle back and say, hey, let's do it even better. So with that, I hand it off to you two gentlemen.
Good afternoon. I think the format that Rich and I decided was that I would start and essentially run through a slightly modified version of the report we provided to the council. Because the report itself has all the details, it's going to hit the highlights, and then I'll pass the baton to Rich, who has more specific familiarity with your process. And if that's okay, we can take questions at the end, although if there's something really pressing, I guess it's okay during, maybe we should try to rattle along through, if that's okay. Yes?
Yeah, I think
Excellent.
that's great. Yeah, okay. We'll take questions at the end, but if you think of something and you need to write it down, you can also throw a question in the chat, and we'll get to it at the end.
Great. And do we decide that you are going to go sort of next slide and control the control
ready?
Things? Go for it. Yeah, so here we are. These are the members. This is available. The report was a couple of years ago. I think I was actually the chair at the time and perhaps the lead instigator of this report, although the committee may have come to regret that fact. Anyway, there we are, ex officios and members. Next. Importantly, ISAB reports are not generally reviewed before they're released, but in this case, because there were a lot of people that we felt had special information, we did reach out. Wasn't a sort of peer review where we were obligated to make all their modifications, but we got a lot of very, very useful feedback. And without calling out which person made which comment, we want to acknowledge the very useful comments we had. In many cases, they were saying attaboy. In some cases, they were saying that we needed to reword or revise, but that's what was all very, very useful. Okay. By the way, there is some outside noise. Am I coming through okay?
We can hear a little bit of
there's some neighbors
like
that are like power washing or something, and I can't control that, but that's rural living. Okay, so the goals, and some of this may be self-evident for some people, but I want to go through it. Why survival and return estimates are so important. You might think you know what a smolt and an adult is, and survival and return, but the terms turn out to be a lot more slippery than you might think. The nature of the problem, some of the issues related to the definitions, like what is a smolt, an adult? What does it mean for fish to survive and to return? These interface with different marking mechanisms because marking approaches are not independent of the kinds of data that they produce. Try to keep out of the weeds, although there are a lot of weeds, and then pass the baton to Rich for some of the efforts to do better. And I'm hoping this guy is going to leave pretty soon. Okay, next slide, please. So the main thing is that there is almost all aspects of salmon conservation and management in the basin, and salmon conservation and management in general rely on survival and return estimates. Comparing hatchery to wild runs, hatchery practices, optimization of smolt size is frequently for survival, growth patterns, dates, off-site releases, whether hydro project operations have been efficacious with respect for salmon. We typically want to know whether they helped, and survival and or return are generally the metrics. Transportation practices.
Tom, could you try and get a little closer, maybe?
Yeah, okay. And I think this guy is gonna.
Hopefully, he will be done in a minute. But
I'm really sorry. This was not planned.
Of course not.
Is that any better? I think he's gonna go. And of course, managing U.S.-Canada treaty fisheries, tribal fisheries. So we need this for a lot of reasons and more. So we can move on. Next, please. Yeah. So just to quote from the report as examples, just examples of how smolt-to-adult return and survival are essentially baked in. Fish and Wildlife Program's goal is to contribute to achieving a smolt-to-adult return ratio in the 2 to 6% range and so forth. Comparative survival study uses that 2 to 6% SAR objective. Lower Snake River Compensation Plan generates these metrics, both of them. And so the ISAB felt that there was sufficient confusion and uncertainty as to how these were generated and how they differed that was necessary to have our report. So next. There are lots of definitions, but the simplest version is smolt-to-adult return is a proportion of smolts that survive all natural and fishing mortality to get back to some designated place. So smolts out to adults back as return, either returned or they didn't. In the case of smolt-to-adult survival, it's those that survive to adulthood. So if they got caught in the ocean or in the river, they still survived, even though relative to some particular location, they did not return. Hypothetically, if there was no fishing at all, return and survival would be essentially the same. And in some cases that's true, and in some cases it's very much not true. And so carelessly saying, oh yeah, I meant to say survival, but really it's just return or vice versa causes confusion because they're not the same. You have to basically add back that ocean catch or river catch for the survival because they survived to be big enough to get caught. We'll get to that complexity later. So next. So this is a construct from my former graduate student, the illustrious McGill professor, Andrew Hendry. And I like it because it applies to a wide variety of situations. So this is the baby, the werewolf, and the silver bullet. So whenever we're thinking about a situation, what's the baby, what's the werewolf, and what's the silver bullet? Have I got your attention? Okay, first, what's the baby? Next one. So the baby is we need good estimates of smolt-to-adult survival and return for management and so forth. Baby is important, okay? So what is the baby vulnerable to? What is the werewolf here? Next. Werewolf is confusion and careless use of the terms. SAS and SAR are different. They're not independent, but they are different. They use different techniques, different assumptions, and each one of them can be calculated in multiple ways and commonly is calculated in multiple ways. And all of those things hinge on what is a smolt and adult, what is survival and return, and those are not nearly as obvious as one might think. And so there's lots and lots of opportunities for confusion. In some cases, it might result in trivial errors, in some cases, really substantial ones. We're not implying that nobody is aware of any of these things, but in our review, it was astonishing, at least to me, and I've been in the salmon business for 50 years, how often these things get mixed up and how much capacity there is to get them mixed up. So not implying that none of the members of this group or anyone else doesn't know all this, but they're clearly published papers that are very confusing individually and collectively. So there's the werewolf. So what's the silver bullet? Next, please. Well, before we get there, the report wades into the weeds. There are lots of weeds. This, of course, is Humphrey Bogart and Katharine Hepburn and the African Queen. Lots of weeds. The report has lots of weeds. We'll try and avoid most of them here today. And then finally, next. Our silver bullet is clarity and consistency. And Rich will really emphasize this, but when terms are used, they should be defined and used consistently. And sliding back and forth only causes confusion. Salmon can be lost in the river before getting to sea, at sea, adults going back up. And rolling them all together is confusing because it mixes up the sources of mortality and it makes it harder to compare to other data sets. And we also suggest that annually reporting smolt-to-adult survival from Cote tag for a set of stocks and both SAR and SAS would be useful. Some of these are already done, for example, from the Chinook Technical Committee, but there are other ones that might be added to that without an onerous burden. And maybe it's not a silver bullet, but at least it's going to stab the werewolf a little bit. Okay, so let's move on. So apples and oranges here. First of the metrics, smolt-to-adult survival and return are both metrics, but they're not the same. They should not be used synonymously. And second, there are three major marking approaches, although in the history of salmon ecology, there are more. The first main one is coded wire tags. Second main one is PIT passive integrated transponder tags. And the third, coming on very, very fast in the last decade, is parentage-based tagging, abbreviated PBT. And the two metrics and the three marking approaches are not independent of each other, as we'll see in the next slide and thereafter. So first thing is neither SAS nor SAR is marine survival, even though a lot of people use them that way. And I'll explain that in a minute. So carelessly calling it marine survival, smolt-to-adult survival does not help. Coded wire tags are usually associated with smolt-to-adult survival because they're heavily sampled in fisheries. PIT tags are, and this is not necessarily, but typically associated with smolt-to-adult return because we can assess returning fish that are not caught, that are still alive. But those pit tags are not typically sampled in marine fisheries. And then we also get, as Rich will elaborate later, lots of combinations. Hatcheries that might have 1 or 2 or even 3 different metrics over different periods of time. So there's a certain amount of overlap. So next slide, please. Coated wire tag, and I know most people are probably very familiar with it, but just to remind folks, primarily applied in hatcheries, not necessarily. There are some cases, for example, here in Puget Sound, where wild coho salmon are tagged routinely, but generally in hatcheries. You can have individual sequential coated wire tags. My group has done that, but it's a huge pain. So they're almost always batch tags, fish from a certain release pond, a date, an average size, rather than individual data. They are widely, although certainly not perfectly, sampled in fisheries. There's a head recovery program, adipose fin clip, and so forth. So their fisheries are set up to be sampled for that. And of course, because of the location of the tag, it's either going to be dead or get killed in order to get the tag back. It's a one and done. The fish has to be in your hands. Next, pit tags, of course, are commonly applied to both hatchery and wild fish from either a point source or when captured en route at some place along the way. Typically, individual data are recorded. It might be the date of tagging, length, mass, some other specific information. So different from coated wire tags. Typically not sampled in fisheries. It's not as though they conceivably couldn't, but it's basically not set up for it. So ocean fisheries are not typically sampling for pit tags. So we don't have an assessment of the number of fish that survived to adulthood but got caught by a trawler off the coast of Southeast Alaska. But they have the advantage of multiple so-called hands-off detection. So that smolt released from a hatchery could be detected multiple times on dams going out to sea, and conceivably multiple times at dams going back upriver, unlike the coated wire tag fish that obviously you couldn't. And that provides some advantages, obviously. The third, parentage-based tagging, yeah, go ahead. Kind of magic to people that aren't geneticists, but by sampling all the adults, fin clips in a hatchery, and not doing anything more, the geneticist can determine all the possible offspring genotypes. And so effectively, all the juveniles from that hatchery are known to that hatchery, to that brood year, and in fact, to their parents genetically. They're not literally tagged, but they are really parentage-based known rather than tagged. So we have families, but not individual fish data. We don't know the date when they were released or their size because we don't put our hands on the juveniles. They're marked genetically. Not typically sampled in fisheries, but you can sample them, let's say, on a spawning ground without killing them, but you have to have your hands on them. You can't detect them remotely as you can a pit tag. So all of these methods have different approaches and different strengths and weaknesses. So next. Here's where it gets tricky. And again, I've been in this business a while. I would have thought I knew these things, but it is not that simple. What is a smolt? What is an adult? And if you're going to have smolt-to-adult survival or return, you need to know what's a smolt and what's an adult. What does it mean to survive and what does it mean to return? And the devil, as always, is in the details. So here we go. Prepare to get confused. You can release a fish from a hatchery and they might call it a smolt, but it might not migrate. It might enter the ocean at different times of the year. It might zip directly down the river, go part way, and then hold for a number of months in some sort of a saltatory migration pattern. So release from a hatchery may be called a smolt, but the physiological state, the migration pattern, the exposure to mortality in the river might be quite different. And studies by some of the NIMS physiologists, Brian Beckman, Don Larson, and others have dug into this, but not every fish moving downstream is a smolt. and not all fish released to smolts move downstream. They don't move downstream in the same manner. In fact, there are considerable variations. The next thing is why a smolt to adult is not the same as marine survival, because, as of course you folks know, Columbia is a big river. And if you're in Idaho, the fish may be a smolt, but he's definitely not entering the ocean. So where does it start? A tributary, lower Granite Dam. If the fish was released from a hatchery farther down, it couldn't be counted at lower Granite Dam. And so its start place would be somewhere else. So the smolt return includes different sections of the river. And then, of course, in many ways, people say, well, you know, Bonneville Dam's the end of the river because that's where we stopped counting. But there's a lot of Columbia River below Bonneville Dam and a lot of salmon, but we don't have pit tag detectors there. So the comparability of a lot of those important stocks on both sides, obviously, is different because we don't have ways to catch them. So where do you start being a smolt is not obvious. The second thing is, next please, out and back. And so when the adult returns, if it was detected with a pit tag at Bonneville Dam, okay, it got to Bonneville Dam. But if it entered a hatchery or someplace below Bonneville Dam, what do we know about it? And we have multiple sources of natural and human-related mortality as the fish works its way back. So the smolt to adult return, it has to have two anchor points. Where it starts as a smolt, that's where we count it, which is not necessarily where the fish started, but where we started it, and then where we brought it back. And it could be lower Granite Dam to lower Granite Dam, lower Granite Dam to Bonneville, Bonneville to Bonneville, Bonneville to lower, or any combination, but it has to be specified. And if we're comparing among hatcheries or hatchery and wild stocks, we have to make sure that we're comparing apples to apples. Otherwise, sources of mortality in the river to juveniles going down, in the ocean, and in the river going back up are all rolled together. We have different kinds of processes. The likely effect of human activities is different in different sections there. And obviously the fisheries and the sampling and so forth are also different. So we strongly advocate as much as possible, particularly where you have things like pit tags, not rolling it all together, but considering or at least reporting some of those life history migration segments. So next, what's an adult? This is my former student, Scott Gendy, catching a Chinook salmon off Juneau, clearly an adult by anybody's standards, right? Okay, good enough. How about the next one? Go ahead. So the gentleman on the left is participating in the Tengu Derby. This is a Japanese-American traditional sport fishery in Puget Sound in the winter. So this is clearly not a smolt, but it won't spawn until the following fall or some fall after that. Is that an adult? It's caught, it was taken in a legal fishery, but it's not the same as a fish that is older and has been exposed to more mortality. And next, please. This is clearly an adult, right? I think anybody would say so. I think Morgan took that in the Willamette's Mackenzie River or some such place. That's an adult. It's returned to spawn. It's all colored up. Yeah, good enough. That's an adult. Okay, how about the next ones? So over on the right, that's my hand for scale. This is a naturally spawned, sexually mature male par. The geneticist would say, well, he's absolutely an adult. He could contribute. In fact, these par often do contribute significantly to natural reproduction, almost entirely unsampled. The other four fish, and there's a Sharpie for scale. I took this photograph back when I was a graduate student. Mini Jack, age 3 and age 4 Chinook salmon at the UW hatchery. They're all sexually mature. They're all adults, but are they comparable? And I think people will say, well, yeah and no. So why not? Next, please. So the problem is that while they all went to sea, except for the par that didn't even do that, they've been exposed to very different durations and therefore levels of natural and fishing mortality. And so if you count all those, the jack, the mini jack, the age 3 and 4 as equivalent, anything you do to increase the proportion of those younger ages at maturity will inflate the apparent survival. And this is not new. People discovered this decades and decades ago as they released larger smolts to have higher survival. In some cases, they simply had more jacks. And that's known, but in many cases, people do not make an adjustment for that fact. And of course, we don't know precisely how much mortality there is for each year at sea. And our report goes into great detail on the difficulty in estimating that, and the different people who've come up with estimates. But in some cases, no adjustment is made for the age at maturity, which varies. Commonly, of course, in Chinook, coho, and steelhead, also sockeye, although that's less pressing in the current situation. So keep going now, please. Here's an example from some of Rich's work comparing hatchery and natural origin males and females. And I've drawn ellipses there so you can see that the hatchery origin males and females both tend to be younger, although all three ages are represented, than the natural origin fish. And so if you simply counted a fish as a fish without any kind of adjustment, you might infer that the hatchery fish have a higher survival. In truth, their survival rate, that is the per year survival, might not be better, but you would just get more back because those fish that were planning on staying out to sea and coming back at age 5 in the natural origin population died trying. And again, some reports, some papers make this adjustment, lots do and lots don't, but in many cases they don't say whether they adjusted or not, or how they adjusted, or what was the basis for that adjustment. Okay, next. In some cases, in addition to the hatchery wild comparison, we are seeing in long-term data sets changes in age composition. I mean, there are a number of papers that have published on this, including Ricker back in the 80s and many others up to the recent period. We are often seeing a reduction in average age, although not necessarily. But if we are, that complicates comparisons on long time series of survival rates if we simply use the number of fish returning or the total survival rather than the rate with some sort of an adjustment. Okay, next, please. So what does it mean to survive to be an adult? And again, think back to that big Chinook held by Scott Gendy, my student, and the smaller one, large but not mature, held by Justin Wong. So in management standpoint, salmon recruit to fisheries. So they get caught off Southeast Alaska. A lot of Columbia River Chinook do, for example. They're considered adults, but it's acknowledged that they might not mature that year. You don't know. And so factors that affect survival at sea often call for an adjustment. So in some cases, some management agencies, like Pacific States, the Chinook Technical Committee, assume 20% mortality per year. And so fish maturing at an older age are essentially upshifted to count for more than one fish. And fish that matured at a younger age than the mode are downshifted. Other people assume different mortality rates. We basically don't know what the at-sea mortality rate is. But some people estimate it differently than others, and some people don't adjust at all. And it's worth bearing that in mind. What you do is your business, but our ISAB report was designed to kind of call to people's attention some of these aspects. So next, what's survival? So typically we would say that if they recruited to fisheries, so there's my friend Wayne Paulson with the Puget Sound coho salmon caught in probably September. So that fish survived. There's a female Chinook in a hatchery. She survived. There's a dead Chinook on the Yakima River. These fish all survived. But they're being sampled at different places. And depending on where we have our assessment, they may or may not be added in. Okay, further, we also have issues of marine fisheries. So these are Southeast Alaska gill netters and trollers, just sort of random pictures I've taken. They're typically sampled for coded wire tags, although sometimes the sampling fraction is pretty small. So one actual recovered fish with a tag may be expanded to 100 or something like that. But the pit tags and the parentage-based tagging fish, the genetic fish, are almost never assessed. So if those salmon stocks are routinely taken in these kinds of fisheries, we won't pick it up. That'll just be rolled in with a natural mortality and won't be distinguished. They actually did survive, they just didn't return. We can't tell the difference. So next, we have return. And so in some cases, if you are detected with your pit tag at Bonneville Dam, you returned. But in other cases, you don't count as having returned until you got to Lower Granite or someplace else. And again, as with the Smolt-to-Adult, we need to point out clearly what are the points where we're calling it a smolt, where did it start its assessed life, and what does it mean to have returned? It's confusing when some fish are returning to Bonneville Dam and counted as having returned, and others aren't counted till they've gone hundreds of kilometers farther upriver, including possible natural mortality from heat stress, pre-spawning mortality, fishing, and whatnot. So we want to be clear, again, how to distinguish fishing and natural mortalities. Next, please. And so in some cases, survival and return are similar. In some cases, they're not. So if they're marked and counted at the same place, so it's a hatchery, released together and not exposed to fisheries. So for example, many steelhead populations and some interior Chinook populations have very little intercepting marine fisheries. Then in those cases, survival and return might be pretty much the same if we counted them back to the same place. But if you have a lot of the lower river fall Chinook salmon that are quite heavily exploited in coastal fisheries, they're not the same. Survivals and returns will be quite different. And this is partly why we have one of our suggestions is to have reported levels of those for a number of stocks and keep track of that, because the degree to which they track will depend on where the fisheries are going, patterns of migration, age at maturity, and so forth. Next, please. At this point, I'm going to turn it over to Rich. He put these together and he will try and lead us out of the weeds back to things that might be most specifically relevant. I appreciate the invitation to speak to you. I hope I haven't dismayed you overly. Over to you, Rich.
Okay, thanks a lot, Tom. I think you laid a good foundation. What I'm going to do with the next 6 or 7 slides is I'm going to cover some past efforts to standardize SARs and SASs, and included in that will be a discussion of the Lower Snake River Compensation Plan hatchery programs and a little about that history. I'll touch on the ad hoc supplementation technical work group that provided guidance for SARs. And then I'll talk more extensively about our review of the coordinated assessments, and then touch on recommendations at the end. So starting with the Lower Snake River Compensation Plan, which was initiated in the mid-1970s, a hatchery program in all the major basins in the Snake, above Lower Granite, and also the Toucan and below. Adult goals were established for each of those basins, including the Imnaha, Grand Ronde, Salmon River, excuse me, Clearwater, Toucan, and then some outside basin mitigation in the Walla Walla. And since there was adult mitigation goals established for those basins, smolt production programs were developed, and there were SAR targets developed for each of the hatchery programs. And those SAR targets were defined very explicitly, basically the return rate you would need from the number of smolts that the program was producing to meet the adult mitigation goal. And in most cases, that was the release location for the smolts, which could have been a hatchery, acclimation facility, or direct stream, and the adult returns to Lower Granite Dam. And then in addition to that, there was an assumption that adults lost due to construction of the dams to downriver fisheries, both ocean and in the Columbia, and the snake below Lower Granite also needed to be compensated for. So there were SAS targets established. And particularly for the Lower Snake Comp program, they got it right pretty much from the start and have, since initiation of these hatchery programs, been tracking their SARs and SASs and comparing them to their goals. Next slide, please. There's been a major transition in methodology, and Tom talked a lot about the advantages and disadvantages of different marking methods. And so this represents the array of approaches that are currently being used within the Lower Snake Comp program. To estimate SARs, there's strictly coded wire tag-based estimates underway. There's parental-based tagging, and there's pit tagging. And for SASs, there's coded wire tagging, parental-based tagging, and then combinations, PBT plus pit, PBT plus CWT, PBT plus pit conversion with coded wire tags included, and then PBT plus coded wire tag surrogates. So the methodology has become quite complex. And of course, there's pros and cons of these changes that have occurred. I think the PBT and pit methods have provided more precise and accurate estimates, particularly for SARs and in many cases SASs. I think one of the major complications that occurs with the changes and advancements in these methods is the comparability of the long-term data sets. Particularly because most of the PBT and pit tag estimates include fish that would have died above the adult return location. For example, if it's Bonneville Dam, it's an estimate of the fish that arrive at Bonneville. If that estimate's based on coded wire tags, it's back calculated down from all the recoveries that occur of those coded wire tags above Bonneville, which doesn't include any natural mortality. So that's just something to be aware of. Next. I'm going to talk a little bit about the ad hoc supplementation monitoring and evaluation work group. The problem that work group aimed to address was the diversity of data collection strategies and analytical approaches employed across the region for hatchery and supplementation evaluation, placed a substantial limit on our ability to combine data across projects and to enable large-scale analysis. And therefore, there was a need for standardized definitions and protocols. And so that was really the major goal and objective for this work group, was to describe a framework with which hatchery monitoring and evaluation might be standardized for long-term and short-term effectiveness. There were 50 standardized performance measures defined and recommended in that document, and SAR was one of those, and it was defined as an important metric with specific methods described for 4 SAR types. And unfortunately, one of the SAR types included incorporation of all harvest in the ocean and in river. And therefore, these recommendations contributed to the confusion because they were, and I should say we because I was an author in this report, we were calling an SAS actually an SAR. So next. So the current efforts to standardize in the Coordinated Assessments Project, everybody knows what the goal of the standardized, of the Coordinated Assessments Project in the CAX. And a little bit about the SAR-DES. It contains 50 fields for characteristics and supporting metrics of each data set, and actually each estimate. And of course, in the ISAB report, we say this complexity is essential and important. And I think Tom highlighted that in his initial presentation there. But it obviously results in many SAR types over a wide range of biological and geographical scales. And the problem that we identified is one of the SAR acceptable options for adult return data definition is fish surviving to adulthood, which includes ocean, mainstem, and tributary harvest in the adult return estimate. So similar to the ad hoc supplementation work group, this specific SAR is actually an SAS estimate and also contributes to the SAR-SAS confusion. Next slide, please. Here's a couple of quotes from the ISAB report. Consistency would be improved in the DES and the data that are shared on the CAX if the difference between SAR and SAS were clearly indicated and SAS was presented as a separate high-level indicator. Another quote, providing both SAR and SAS would be extremely valuable for a variety of comparative analyses to better facilitate local and regional management decisions. So solving the problem, I don't think is all that difficult, although I think the protocol and the process looks like it's way more challenging than solving the problem to me. The current SAR-DES has all the required data and metadata to calculate and support the SAS estimates. And I don't think there's a lot of problems with the existing natural origin data that's posted in the CAX because there's very few of those data sets or any that have the ocean harvest included. And so I think most all of those data sets are actually SAR, and there may not even be any SAS data sets in there. Next slide, please. The other thing that I'd like to have the group consider is creating the ability to generate these catch and escapement distribution profiles from the SAS data sets as they're posted. And I believe all of the data you need to generate these is already captured in the DES. In some cases, it may not be captured at this scale for strays, for example, or for harvest. But in the review of the Lower Snake River Comp Plan program and this ISAB report, the ISRP and ISAB members really found tremendous value in these catch and escapement distribution profiles that are very information rich. And if there was a way that these profiles could be extracted along with the SAR and SAS data, it would be really valuable. It'd be a great contribution for the CAX if this information could be extracted. Next. I didn't want to leave the coordinated assessments or this group with the idea that all we did in our report was criticize or make recommendations for improvement. There's strong support for the coordinated assessments project, and I think that support is highlighted in the ISAB report. For example, you'll find the statement, the CAP project and the CAX have greatly improved the quality, quantity, and availability of important high-level indicator data for natural and hatchery origin salmonids in the Columbia Basin, and rigorous documentation requirements for all data sets, including methods, metrics, and metadata, are highly beneficial for assessing whether specific data sets are suitable for comparative analysis. So we're strongly supportive of the effort. And the other thing I'd like to say is I think the ultimate value in a report like this is when people actually use it and make changes that are beneficial. And I think we're happy and proud that this group is taking the recommendation and trying to make some changes that I think might really provide a lot of benefits in the long term to the data that's shared. Next slide, please. Just to highlight the 7 recommendations, these are summary recommendations from the report, and they reiterate some of the things Tom already presented. So first recommendation, to provide clear objectives and applications for studies that use SAR and SAS. Define SAR and SAS and related terms clearly and use them consistently. Provide clear descriptions of how the metrics and terms are related and estimated. Augment SAR reporting and publicly accessible databases to include SAS, and component life stage specific survival where possible. And the first part of this, of course, relates directly to our recommendations to the Coordinated Assessments Project. Next, please. Recommendation 5 is to maintain the integrity of the long-term SAR and SAS data sets by comparing results of different marking and analytical methods and developing robust conversions. And really, when I say different marking and analytical, it's the long-term data sets in the basin that are based on coded wire tags versus the newer data sets that are based on either PIT, PBT, or those hybrid combination methods. And much of this work has already been done in the last couple reviews we did of the lower snake. Both IDFG and ODFW did some comparative analysis that was very informative. And so I think sort of formalizing that and making it a little broader in scope and sharing the results of that would be very valuable. Where appropriate, as Tom pointed out, there are situations where adjustment of SAR and SAS estimates to a common age is useful for comparisons. And be sure when that happens to explain any adjustments, because that's not always been the case. And then be cautious when using surrogates and provide clear explanations of how well the surrogates represent the population of interest. Next, please. So the world of SAR and SAS, as Tom pointed out, is a weedy place. And the report explores the weeds, but the key is to address those recommendations that I just presented as appropriate for whatever the task or the study is that's at hand. Last slide. So we'll take questions here, but again, I want to really thank Mari and Casey and everybody else that invited us to come and present and to actually consider the recommendations that we provided. I think in the long term, if we can make the kind of changes that we suggested, I think the databases are going to be more widely used and more valuable in the long term.
If I might just add one quick thing at the end to reinforce what Rich said. The goal here is not to sort of scold and tell people they've been doing things wrong, and the goal is not to tell them how they should do it, but just to point out some of the considerations. And we hope that the report will be helpful in allowing each group to decide what metric and what form of adjustment and analysis of the data is most appropriate for them and move forward. And I know he and I have both phased out of the ISAB, but would be happy to communicate with anybody that wanted any further information from us. The goal is to be helpful. Thanks for inviting us.
Yeah, thank the both of you for joining us and giving a great presentation. And I agree wholeheartedly in if someone uses your resource and then says, it's great, here's how you can make it even better. That is the investment that means coordinated assessments has survived for the last 15 years and will in the future. And we've always tried to be as adaptive as necessary as we saw things more clearly. I have not kept up with the chat. Are there any questions in chat, or do folks want to start raising hands? Time for questions.
You're all good on chat right now.
Okay.
But if you guys have questions, feel free to put them in the chat or unmute yourselves. Don't be shy. We have Rich and Tom here. The best possible way.
Yep.
Not a question, but this is Mike Bannock. Rich, you are correct. I looked at the data that we have here, and ocean harvest is null for all of them, except for one steelhead population where it's just all zeros. So
yeah, and so I think as far as adjustments to the natural origin SAR data, the actual databases, the database and the individual estimates, there's probably very little that needs to be done. You might even adjust the DES and the data stays the same. You just remove potentially some of the fields or the metrics, and it becomes an appropriate SAR DES.
Good. Mike, do you want to turn on your camera and introduce yourself? You weren't here when we did task team member introductions.
Sure. Hi, I'm Mike Bannock with Pacific States Marine Fisheries Commission and database manager for StreamNet.
Thanks.
Sorry I was late. My computer didn't tell me 2 hours ahead of time like normal.
Okay, the floor is open. Brian, thank you.
Yeah, so I guess I would just agree with one of Rich's early comments about I think there's some really low-hanging fruit and some simple changes or modifications to the existing DS to capture these things. One being to specifically have an SAR and SAS estimate. I think that's pretty straightforward. And then just making very clear about the definitions on the locations that you're measuring and the adults that you're including. And then I think there's already a kind of with and without jack kind of stuff in there, and that might have to modify it a little bit for other species than Chinook, but I think there's some fairly straightforward and simple things that would create a lot of clarity to the existing DES.
Thanks, Brian. Bob?
Yeah, it sounds like I can just go ahead and introduce this one right now. Having been peripherally involved in the writing of that report as an ex officio on the ISAB, I noticed that even, and this is a kudos to everyone for falling into the same trap. Even in the writing of that report, we were encountering some of the same language barriers. And I just heard the word used again, and it was my biggest pet peeve and has been for a decade or more. It's the usage of the word include, right? So we all know that a SAS and a SAR, one could be forward calculated to be the other can be back calculated to be the former. And when you say something like a SAR that includes everything that made it back to the spawning ground, but you're implying that it's reintroduced into the number to get a SAS, then include basically doesn't mean include. It means had made it, but then is being removed and re-included into it. And I'm doing it right now. We have to spend some time on that one word specifically, because the number of times I've realized, okay, how many times do I have to read this until I figure out what that one thing means? So I think there's a missing word that can supersede that one that seems to fall apart going forward, but works pretty well going backwards. And so that's my comment. It's a single word comment. We got to get rid of the word include and find a better one.
Hi, Bob. I happen to agree. In fact, as I was going through this, trying to read and speak at the same time, the very thought crossed my mind. You're absolutely right. We have to be careful of that. If the fish is caught at sea, we know in terms of survival that fish counts. Whether you call it included or excluded, put in, taken out, is sometimes ambiguous. And that's another part that should be clarified. What we exactly mean. I mean, I think you and I understand the same thing. It didn't return if it got caught at sea, but it survived. But how we use that word, and as we read the methods sections of papers and reports, it's often not really clear. So absolutely right, I agree.
Yeah, thanks, Bob. I have also struggled with the word include and adjusted. And so I'm happy to hear I'm not the only one and agree that, yeah, if we can find some better language, have some examples about what we mean when we say include, or just, what that exactly means. Yeah, totally agree. Mike, I see your hand up.
That was actually going to be the same thing that I was going to say. Since the day we first made this table, that what does include mean? It's one way or the other, and I've never been able to quite understand. But I did want to ask you, Rich, it sounded like you're, other than this include word, and it sounded like you would, maybe you'd characterize what we have here as like all the pieces are there. Maybe we just need some relabeling. We just need to kind of just change the language a little bit. Just, it's like, here's what this really is. Here's how we should reword things. Is that fair to say?
I think so. I went through in detail that DES and looked at every one of the pieces of data that were requested or went into the entry to the calculation, and I think everything was there, that you needed. I'd have to go back and look to see if that was the case to generate the catch escapement distribution profile. But I think it was all there. And I'm going to backtrack a little. I do agree with the problem with include, and I think part of that comes from different methodologies. For example, when you use coded wire tags and you add up everything that's recovered above a specific location like Lower Granite Dam or Lower Bonneville, those are all added up and included in the estimate. But then when you do a pit tag or a PBT estimate at a dam, that's just an estimate of that number of fish, but it also includes the fate of every one of those fish that would occur after that time frame above that location. And so that's where it gets really kind of complicated with the inclusion and not inclusion. And I used the natural mortality as example because I think we have to be cautious in comparing, for example, old CWT estimates of SARs with new PBT and PIT, because the new estimates have natural mortality as a component added in, even though you're not adding it in, it is, those fish are in that estimate, where the CWT methods never have that in there. So that was the point I was trying to make there with, and I think that weaves together with the inclusion problem.
Thanks.
Tim, go ahead.
Yeah, I guess I wanted to comment that a lot of this is driven by the question at hand and where you can make the measurements that help you address that question. And you could calculate, you know, you could calculate an SAR and SAS in lots of different ways, depending on what is you're trying to address and how you can make the measurements that you need to make. Like for instance, a lot of the goals we have for populations are in terms of adults, which don't include jacks or mini jacks or par. So those things don't count for that question, but you might, if you're managing a stock for a fishery, then you know maybe you would count those because you may be harvesting fish in the ocean. So same stock, but different questions. And I think the clarity needs to extend to that. And I'm kind of wondering how changing the DES, I mean, is that really going to solve that problem? Not that it, you know, might not help, but I don't know that we're going to completely solve that problem.
So, you know, I totally agree with you, Tim, and I think we make a pretty big point of that in the report, and even that last slide that Tom had included that I presented, which was the task at hand. The appropriate method for the appropriate objective is really important. Possibly you could add something in the DES, but my thoughts are the method descriptions that are connected with the DES should have some explicit description of what the purpose of those particular estimates are and how they're being used. And if that's not the case, maybe that's something you would want to add in the documentation. What's the management application question, research application or question? But I think that's really important, Tim, for understanding why the particular scientists chose the approach and the metrics that they chose specifically to answer the questions they're trying to address.
I often say about coordinated assessments and the work that we do that it will never be perfect, but we can keep improving. And that as long as we are willing to hear possible improvements and apply those as we best can at the time, then we continue to, as Evan would say, better data faster.
I think Tom made this point, and I think I'm just going to reiterate it. In some cases, a lot of the problems we observed and the recommendations we made don't apply necessarily to people like and programs like Brian Leth in the lower snake evaluation in Idaho, or Joseph Feldhaus in the evaluations in Oregon. A lot of this arose from other people going and taking other people's data and not understanding what it was and doing analysis and coming up with conclusions that just were not sound. They weren't scientifically sound because they didn't understand the definitions and the assumptions and the nuances of the data. That's not to say we can't improve how we communicate even some of the best data from the best managed programs. We should make sure we're doing a really good job of that also.
Yeah.
Yeah, I agree. There's many people that know all that they need to know and are doing things absolutely correctly, but that doesn't always stop other people from grabbing a line from their paper or a table from their report and folding it into some multi-stock analysis. And then the newspapers or the public sees that and says, oh my God, what's going on here? And it takes a lot of time to backtrack. So just seconding Rich's point, reaction here.
Yeah, thanks, Rich. That's a great point. And, in providing this data publicly, it's really important that we're careful in how we describe it and how it's defined, so that it is used correctly. So I really appreciate that and appreciated your report and helping us, get that data, the SAR, SAS data described correctly. I know it's something that we've been thinking about for years and just really needed someone to kickstart that process. So this paper was that kickstart. So
yeah, and I can't even remember, I retired 11 years ago. I can't remember how much. I know I had a lot of input in the natural origin abundance and productivity recruits per spawner adult to adult. I can't remember how far along we were in the SAR, but if we were near the end when I left, I apologize for not catching this back then, because we could have saved us all a lot of problems, right?
We've got data that folks can look at and say, wait a minute, could we describe this slightly better? Right? So I don't think that this is a big stumbling block. I see it as investment in what we've already done. I had not realized that it was 11 years ago.
Yeah. A quick question, and this would be feedback from the group. What do they think a priority would be on being able to produce those catch to escapement distribution profiles? How valuable would that be, and how difficult would it be? And maybe Tim Copeland, Brian Left, I don't know if Joseph's out there, maybe Bob Lassard, whoever works in that arena, what do they think about that?
Go ahead, Brian.
Well, from my understanding, and I've been somewhat on the periphery on the DES, and we really just started working on getting hatchery data in there. But from what I can understand, none of that is really possible at this point. I don't think I think there might be some draft tables for some of the harvest information, but I'm pretty sure there's not like a completed DES that you can upload harvest data into. Is that correct?
That is correct, Brian. But a bit of a snippet, somebody has maybe been able to complete the circle between coordinated assessments data and data in ARMIS. So here's the encouragement for everyone submitting hatchery data to the coordinated assessments. If you put your ARMIS return and release codes in, or your release codes, there is a link to harvest data through ARMIS now.
Well, I do know the DES does have fields for various harvest components. I don't know if it's all of them. I can't recollect if it has fields for strays or not, but it would be valuable, because those are all values that have to go into an SAR or an SAS calculation.
Yeah, so I know that we included them in the natural origin. I'm not sure that they were included in the hatchery origin DESs, but for the natural origin, we had based so much of that off of what the Interior Columbia TRT had done. And Tom Cooney had consumed different methodologies for some of that calculation that were coming out of the Science Center. And then those folks started retiring, and there wasn't continuation of that. And so we have been kind of the hatchery or the harvest data fields in natural origin for sure have kind of been put in that on hold until we have the oomph to do that. because just having them associated with individual population level returns per year, that data isn't always connected in a straightforward one-to-one. Not that became reproducible after there were some losses in steps and calculations from the Science Center in the way that they modified harvest reporting and made that available. But it is on the list of, we want to.
I guess the other thing I was going to mention too, kind of as part of your response to your question, Rich, as far as the difficulty as you became aware through the program review for Lower Snake, that so for some of our programs, when we're looking, because a minute ago you said that harvest is necessary for creating the SAS estimate, and really for the way we do it, the only place that's really true is like downstream of Bonneville. But for all of the harvest that occurs, well, for all of the loss that occurs upstream of Bonneville, that's based on the PVT and pit tag information, and we don't actually enter in specific harvest estimates in that reach. So it's like you mentioned earlier, it's a combined, it's essentially it's an estimate of all loss, harvest and natural mortality and straying together. They're not listed separately. So entering that in that DS would be problematic based on our methodology.
But that would be true for the spring Chinook and Steelhead, but obviously not for the fall Chinook, which would include, I presume, mostly Cote d'Ivoire tag based, and then other agencies. And more broadly, there's still going to be the CWT estimates, which would have the data to be able to do those profiles.
I'm not saying you couldn't do them. I was just saying, like, consistency, there wouldn't be consistent across different methodologies and potentially species and areas.
Yeah, I understand. And maybe it's a whole separate discussion and a whole different future DES or something. I was just looking to capitalize on the efforts that are underway now, thinking that what I had seen in natural origin SAR DES had most of the harvest metrics or numbers available to be entered into the DES.
I'll say I'm not sure what percentage of entry we have for the data sets in coordinated assessments on that. We did include fields, but they haven't. But if they're not required, there is a varying entry for those.
Right. They may not be populated.
Yeah. Maybe Mike can pull up a number quick.
I'm working on it right now.
And for folks who can't see the chat, Tim Copeland said a matrix of fates would be very useful.
Mm-hmm.
I think one of the things with the natural origin SARs, they're principally sort of two types. They're the CSS data sets that don't have any harvest information, pit tag base, or their abundance base smolts out of a population and adults back. So my guess is that most of those wouldn't have any harvest data, but I thought I saw fields in there that were available to enter that data. I guess I didn't.
Hopes and dreams.
I guess I didn't realize that the hatchery SAR DES was simplified from the natural origin SAR DES.
Okay, so here's some summary information for you. Ocean harvest, like I mentioned earlier, there's just one steelhead population that has ocean harvest in there, and they're all zeros. So that's just most likely an assumption. The other two fields we have for harvest is main stem harvest and trib harvest. No records include main stem harvest. For the trib harvest, there's only 87 records, and looks like that's just few Northeast Oregon populations. So that's 87 rows, and and that's out of over 2,000 records. So,
refresh my memory, was there fields where you enter strays?
Okay, that's the other thing I was going to bring up here. So for the natural populations, there's the 3 harvest ones: ocean harvest, main stem harvest, trib harvest. There's nothing in that one about strays. For the hatchery DES, when we went there, because when we made these things, we were already aware of the problem, especially that word we were discussing earlier for the harvest. We didn't include harvests in the hatchery one. We said we're just going to hold off until we kind of get this figured out. So hopefully this process we're doing right now is figured out. The hatchery one does have a field for strays removed, but that's not which of these fish went somewhere else. It's which fish from other, how many fish from other places came to this return spot that we have, and were those fish identified and removed in the SAR calculation.
So if there's strays recovered for a hatchery group of fish that an SAR is estimated for, those are not included?
If there's strays in a hatchery SAR that they can identify as having come from somewhere else. So if you have a Dvorak, if you're working on a Dvorak return and you can tell it's a fish from somewhere other than Dvorak, and it's like, yeah, and if those were removed, we have a field to say those fish were removed.
Yeah. So I was just curious if, for example, Lallastock steelhead, a group of coated wire tagged fish, had a bunch of fish recovered in the chutes. How are they rolled into? Well, I don't want to get in the weeds here, but
we don't. Yeah, if they're removed from one, are they added to the other because they did survive?
Yeah, they need to be, or you don't have an SAR.
That's like collaborative coordination at a regional scale, Rich.
Well, you don't have an SAS, right? Instead of an SAR, but it's one directional. So in this hatchery return record, what did you do with, did you deal with strays? Yes or no? The fish coming back to this spot that you're analyzing, if part of that cohort ended up in other places, we don't have that in the DES.
Well, it could be important for an SAR also, depending on where the location of the SAR is. If it's at Bonneville and fish go into the chutes, then they're recovered there and estimated, they would need to be added into the estimate of the R at Bonneville. And sorry, Bob, for using the word included. Sorry to get into the weeds.
No, this is really good discussion. So yeah, I appreciate it. Does anybody else have any more comments or questions? We've got some stuff going on in the chat, which is great, and we can keep track of it. All right. Do we want to move on to wrapping up, Maury? Okay. So we're just going to wrap up here. And again, big thanks to Rich and Tom for the excellent presentation and your discussion here. And so for wrap up, I just want to say that the task team leads, Mari and I, with help from Meg, will compile some notes. We've got the recording, questions, suggestions for kind of recap and discussion at the second meeting. The kickoff meeting and recording materials will be available on the PNAMP Fish Monitoring Workgroup website and the YouTube channel. And if you want to be involved in our next meeting, or you want to indicate your interest in joining the SAR SAS team, I made a form. There's a QR code and a link. We're looking to get meeting 2 kicked off in about 2 weeks. And I have a timeline on the next slide. You don't have to. Okay. So can you go back to the other slide real quick, Mari? I just was going to finish up. So prior to the next team meeting, we are hoping to have everybody that wants to be involved review the SAR data in coordinated assessments and review the DES so that you're really familiar with what's in there. Review this presentation and the ISAB recommendations. Think about everything we've talked about in this discussion today, and then come prepared to engage and discuss. Mari and I will have some background information put together. We'll have some examples and some suggestions, and so we'll have some stuff prompted and ready to go for that next meeting. Okay, go ahead, Mari. And so just to give everybody a little bit of an idea of what we're thinking for a timeline, we're looking at holding meeting number 2, June 9th, 10th, or 11th. So fill out the survey to indicate your availability or your task team interest. We're going to close the survey down about June 1st so that we can pick the date and kind of get the meeting going. And then we're looking at another meeting like the week of June 22nd or June 29th. We'll discuss that at the end of meeting 2, and then schedule meetings from there on out as needed. Our timeline is kind of quick. We're hoping to present recommendations to the full DES development team by the end of July, or a special session afterward if necessary. And then once we present this to the DES development team, if there are anything that needs to be addressed or questions or concerns from the development team, we will reconvene this task team and try and answer those questions or modify. And then as part of the next step in the process of DES revision is the DDT, the DES development team, will present the recommended changes to the StreamNet Steering Committee in December at the StreamNet Steering Committee meeting. And as Mari said, there's a close relationship between the DES development team and the steering committee. A lot of us either sit on both or observe on both. So there shouldn't be any surprises there. So that is our kind of our schedule. So I think for meeting 2, what we're hoping to, go ahead, Mari.
I was going to say, I don't know that I caught you saying this if you did. We're considering these next meetings on the 2-hour time scale. That seems to be gets far enough into the work to get things done, but it is also something that folks can fit into a work schedule. So 2 hours on these meetings is the plan.
Thank you. I did not say that. Oh, Jake says the steering committee is early November. Thanks, Jake.
Oh, the XCOM is December. Thank you, Jake.
Okay. Yeah.
For those of us that attend all of them, it just becomes a little jumbled months ahead.
So yeah, we're really hoping to take these recommendations and adjust both the natural origin, hatchery origin data exchange standards to fill in these recommendations from the ISAB report. Make sure that when you are sharing that data through coordinated assessments, you're sharing what you think you're sharing, and people are getting what they think they're getting.
Yeah.
And yeah, please, if
questions at this point for the team or otherwise, I really appreciate everybody that showed up today. And again, special thank you to both Rich and Tom for taking the time to really invest in the data for the region.
Yes,
of course.
You're most welcome. Most welcome.
Good.
I want to thank Mari and Casey and the group at the coordinated assessments. This was a very responsive meeting to a request or some recommendations by ISAB. If I haven't been involved in the last 10 years to help improve things, that's on me. I'm here now. I hope I can help.
Awesome. I'm glad that you're able to join us.
Yeah,
you and
definitely.
Pete can duke it out, right?
Well, it was great to see many of you again, and thanks for the opportunity. It was enjoyable.
Good. It's really nice to work with you again, Rich.
Thank you, Tom and Rich. Thanks for your time.
I'll sign off. Bye, everybody.
Thank you, Tom. Bye.